So basically what interphase is after the chromosome replication, each chromosome is composed of two sister chromatids and the centrosomes have replicated. So the pink mass that's in the photo is actually a large mass of chromosomes. And then it's not really visible in this photo, but I think the darker pink dots that are on the side are the sister chromatids. So that is interphase. Now we're going to talk about prophase, and prophase is when the chromosomes condense and the mitotic spindles begin to form. We can see that the gray mass that's protruding from the chromosomes in the center are actually the mitotic spindles. They are the early mitotic spindles that have began to form. They are actually attached to the sister chromatids that are on the end of the cell. During the next two slides, we're going to examine what prometaphase is. Prometaphase is when the nuclear envelope breaks down and the spindle fibers contact chromosomes to the kinetochore. In the first photo, we were able to see that the chromosomes have escaped from the nuclear envelope, that the kinetochore is a slight green color. It is very fluorescent looking. In the second photo, we were able to see that the chromosomes have very highly protruded from the nuclear envelope. The kinetochore is very visible, it is very spindle-like, and the ends actually branch off from the chromosomes and the chromatids that were on the end of the cell. As you can see, now we're going to talk about metaphase, and metaphase is when the chromosomes that have completely migrated to the middle of the cell, we see that the chromosomes that were once broken out of the nuclear envelope have now moved to the middle of the cell in total. As you can see, the black masses are the chromosomes, and they are all located in the center of the cell. In the next slide, we will see the blue mass is actually all the chromosomes, and they are all in the center of the cell this time. The kinetochore is still there, but it is not as important, I guess. It's not as, all the chromosomes aren't as located inside the kinetochore. And the second photo to the right of the same one, we're able to see that the chromosomes are in the center of the cell. In total, they are completely migrated there. There are none spread out anywhere else. Now we're going to talk about anaphase. And anaphase is when the sister chromatids separate and the chromosomes are pulled to the opposite poles of the cell. As you can see in this photo, in the center of the cell, the purple masses are separated from each other. They are not connected this time. That is the chromosomes that have sister chromatids that are pulled apart. And this photo that we see that the chromosomes have been pulled to opposite sides of the cell, it almost appears as if they're reaching out to each other, but they're actually pulled apart, and the sister chromatids have separated to form two different cells, but there is a chromosome from the kinetochore that is allowing them to be connected still. So now we're going to talk about the next to the last process, which is telophase. And telophase is when the nuclear envelope reforms and the spindle apparatus disintegrates. So we see here that the spindle apparatus has disintegrated. The nuclear envelope has reformed. We're able to see that the cells are still attached to each other, but the chromosomes are located in one central area. Now we will talk about cell division, and cell division is when two daughter cells have formed. We will see in this photo that each cell has their own set of chromosomes, their own sister chromatids. They have their own nucleus within the cell. Each cell is independent of each other. We will see that each one has a cell wall. Each one has the ability to later on divide and replicate again. It is important to notice that the cells are not really connected by any chromatids or kinetochore or chromosomes.